Synthesized DNA has revolutionized many fields, such as therapeutics and data storage. Even though reading sequences of DNA is an important step, writing desired sequences of DNA is of utmost importance. In our lab, we are conducting research with the aim of increasing the efficiency of DNA synthesis by using various methods. We are working on building desired DNA sequences. This program can be used to transmit information. When the printer data is retrieved, the sequence for synthesis is determined and a signal is sent to the drive. This information is used to generate the A, G, T, C base sequences for the printhead nozzle. This is then sent to a designated location. Using a printer as a synthesis platform has the advantage of conserving and minimizing solutions used, which are in the size of a very small droplet. It is also very efficient because many types of sequences can be synthesized in a small substrate. The DNA printer utilizes the same approach as the traditional phosphoramidite-based DNA synthesis. After a deblocking process using acid to remove the protecting DMT group, ink containing the phosphoramidite monomer is jetted into each desired site and stacked one by one. First, to prepare the slide for DNA synthesis, drill holes for the inlet and outlet. Then, wash the surface of the recently drilled glass substrate. After that, apply a silane coating which serves as a binding site for synthesized DNA on the glass surface. Afterwards, assemble the functionalized glass slides into a flow cell through which reagents needed for DNA synthesis can go into. This method of photochemistry-based DNA synthesis has the advantage of being cost-effective, simple, and fast. Using photomasks may be inconvenient because they would have to be replaced after each synthesis cycle for parallel-based DNA synthesis, leading to increased cost and time. We therefore developed an optical system using a digital micromirror device, or DMD, that can quickly and accurately synthesize DNA without the need to replace the physical photomask for each cycle. The DMD screen operates like a photomask when the desired pattern is loaded for each cycle, thereby selectively irradiating UV light to the flow cell. Synthesized results can be verified using a microarray scanner. Our lab is also researching spatial transcriptomics. Spatial transcriptomics is a study that can analyze cellular heterogeneity by examining differences in transcriptome expression levels. Spatial transcriptomics analysis can identify cell-to-cell interactions and gene expression patterns, including the location of cells. To perform this analysis, DNA sequences with location information are used as barcodes and then fixated onto a substrate. Intact tissue sections are then analyzed for mRNA, including location information. Therefore, mRNA capture efficiency is crucial. Our current research is focused on increasing mRNA capture efficiency and reducing analysis costs. A 3D microwell array and microfluidic channel were utilized to place barcodes with more types of spatial information than previously possible in limited spots. This approach has the potential to improve capture efficiency by increasing the number of capture mRNAs. Subsequently, the mRNA can be isolated and the transcriptome can be analyzed using next-generation sequencing. The presence of a barcode containing location information enables the identification of the expressed transcriptome at each location within the tissue. We are also conducting drug delivery research using localized electroporation to effectively deliver various substances, such as pDNA, into cells. This method enables simultaneous delivery of multiple substances, which is difficult to achieve with traditional chemical methods. The aim of our approach is to address the issue of low cell viability that arises in bulk electroporation. Additionally, we aim to enable real-time analysis, which has been challenging with PC membranes, by culturing cells on thin pores of 300 nanometers. Our research is based on integrated studies on membrane production using the semiconductor manufacturing process, cell culture, and electrical circuits for electroporation. The integration of these various disciplines is anticipated to aid our research in surpassing current technological limitations and innovating new technologies and applications. During the process of creating membrane chips for cell electroporation, the GXR coating process is used with a spin coater. To ensure a uniform coating, it is important to carefully remove any dust or foreign matter from the wafer. The coated wafer is baked on a hot plate for 1 minute and 30 seconds. A photomask is placed on the wafer and exposed to UV light for 9 seconds. After the development process, exposed and unexposed areas are selectively removed and washed thoroughly. This process utilized an O2 plasma device to form surface hydroxyl or OH groups for the etching process. The vacuum time and power can be adjusted depending on the intended purpose of the process. The following step involves creating pores in the glass used for membrane chips by means of sandblasting. This is a process for seeding the completed chip using cell culture. This is the process of electroporation that occurs after cells attach to the membrane.